Hello everyone. Today we are going to continue the chapter number 5, molecular basis of inheritance, and this will be going to be your theory part 10, and in this we will cover the section NCERT section 5.9, 5.9.1 and 5.9.2. So basically what we are gonna explore in today's class, learn in today's class, it will be about human genome project. So in this whole chapter what we have studied till now, that what is DNA, what is RNA and all those things, and how people got to know that DNA is a genetic material. Then we got to know about the transcription process, translation process, everything that is required in transcription and translation. What is the difference of transcription and translation in prokaryote and eukaryote? Okay, after this, now what we are today going to understand is the Human Genome Project. Let's start. Disclaimer. So, what are going to be today's learning objectives? So, today's learning objectives are: first, we will understand the HGP's goal, timeline, and achievements. Like, what is here HGP's Human Genome Project? So, what we are gonna understand in today's class, that what were the goals of this HGP, what is the timeline between this HGP happen, and what we have achieved from this Human Genome Project. Then we will know the key features of human genome, what is its size, gene count, repeative sequence, and many more things. Then we will understand we will learn the applications of human genome project in medicine, biotechnology, and evolution. Then we recognize the future challenges in genomic research. So let's go ahead. So, today's index is like this. We are going to cover the NCERT section 5.9, which is the Human Genome Project, and in which we are going to cover its two subpart, that is salient feature of the human genome, that is 5.9.1, and then the application and the what are the future challenges, which is 5.9.2 of NCERT section. Then at last, what we will do after we will study these parts, these sections, then we will give a summary where we will understand, what are the different concepts we have studied in today's class. Then there will be some NCERT based questions that we're going to solve, followed by keynotes, and last we will end it with interesting questions which you guys going to solve yourself. And after this slide, what we do usually, we always do, that we will solve the TPP for this particular section. So let's start with today's class. First, we will understand that what was the background of Human Genome Project. So we will talk about the background. There's, so what we have studied, what all we have studied in this chapter, from that we got to know that the sequence of bases in DNA determines the genetic information of an organism. So whatever the sequence of bases is present in DNA, that is going to determine that what is the genetic information of that person, of that organism. An organism's genetic makeup lies in its DNA sequence. So it's all about DNA sequence. So what kind of genetic makeup that person has, it all lies in the DNA sequence. Difference between individuals are reflected in difference in their DNA sequence, at least in some places. So like how we can differentiate between the individual, that is also differentiated between the, like that we can also decide on the basis of the difference in their DNA sequence, at least in some places. And this is the symbol of Human Genome Project. So this understanding led to the desire to determine the complete DNA sequence of the human genome. So when we got to know that it's all lies in the DNA sequence, okay, basis of the DNA sequence, then it became important, it became a desire that we have to now determine the complete DNA sequence of human genome. Okay. Genetic engineering techniques, isolating and cloning DNA, and faster DNA sequencing method made the Human Genome Project possible. So, in the chapter of biotechnology, before this we have did that chapter. In that chapter, we have studied that how this genetic engineering techniques we can, isolate and clone the DNA. So these are the reasons by which this Human Genome Project became possible. The Human Genome Project was launched in 1990. So, if this is important thing, we have to remember what was the timeline of Human Genome Project. So, the Human Genome Project was launched in 1990. Human Genome Project, HGP, a mega project. From this portion, you guys can answer NCERT exercise question number 12. Now, we will understand the features of the HGP. What was the scale and cost? So, we know that the human genome size is approx 3 into 10 to the power 9 base pairs. And estimated sequencing cost initially is $3 per base pair. So the cost of sequencing is what? $3 per base pair. So what is the total estimated cost? So the total estimated cost is approx 9 billion US dollars. This is a very huge amount. Now, if we talk about the data storage challenge, so storing genome sequence in book as type form means if we will like store the genome sequence in book, if we will start typing like A, G, T, G, these spaces in the book, so what will happen? 1,000 letters per page means on a page there will be 1,000 letters, and in one book there will be 1,000 pages. So you understand, in one book there will be 1,000 pages, and on each every page of that book there will be 1,000 letters per page. So by this, we would require approx 33,000 books to store the data from one human cell. So just imagine that we need approx 33,000 books to store the data from like only one human cell, and in that 3,300 books, what we will need in that 3,300 book, there will be in each book there will be 1,000 pages, and on each pages there will be 1,000 letter. So you and that is only for one human cell. So, what are the computational needs? Enormous data volume necessitated high speed computational device. Because we have enormous data, they needed very high speed computational devices. It became very essential. Why it is very essential? It is essential for data storage, retrieval, and analysis of those sequences. Now, if we talk about the bioinformatics development, HGP heavily drove the rapid development of bioinformatics. So what happens? So bioinformatics, if you want to understand in a very layman language, bioinformatics is like that when we combine biology with informatics, when we combine biology with coding, all those things. That is what bioinformatics. Uh, yeah, so HGP heavily drove this rapid development of bioinformatics. So what are now we will talk about that what are the goals of this human genome project? What are the different goals of this human genome project? First was to identify all human genes, discover the location and function of all approximately 20,000 to 25,000 genes in human DNA. So their first goal was that they have to identify all human genes. They have to discover like what are the location and function of all those gene in human DNA. And what was the like number? It is like 20,000 to 25,000 genes in human DNA. Then after, what are the other goals? We have to sequence the human genome, determine the exact order of the 3 billion base pair in human DNA. So we have to determine that what is the exact order of this 3 billion base pairs in human DNA. Then we have to create the databases of what? So the databases are the places base like where they will store the sequence information in a very easy accessible databases. Then develop better analysis tools, improve the software and techniques for analyzing genomic data. Then transfer technology, applied technologies developed during the project to other field also. So here what we have talked about, here we have talked about what are the goals of human genome project. First goal was what? First goal was to identify all the human genes. What are their location, what are their function, every approximately 20,000 to 25,000 genes they have to identify. Okay. Then what they have to do, they have to sequence this human genome. They have to determine that what is the order of this 3 billion base pairs which is present in human DNA. Then they have to create database. So what is a database? So database is an organized collection of data which is stored and accessed electronically. Okay. So they have to create database of this sequencing which is easily accessible. That's why we are saying that they have to make the databases, which is the organized collection of data which is stored and can be accessed easily electronically. Now, then they have to develop the better analysis tool. Definitely, even if they have the sequencing, they have to, even after collecting a lot of data, we have to analyze the data. So they should have the improved software and techniques to analyze that genomic data. Then transfer technology means after even analyzing the data, they have everything. They got to know about every gene, everything, like what are the exact order of this 3 billion base pairs in human DNA. Then they have to apply this technology to develop many projects and many innovations to other field also. So these are the goals of human genome project. Now all like other goals too. Now we will talk about the other goals. First is address ethical, legal and social issue. Ethics, if we talk about ethics, what moral principles might be violated or challenged by the project? So they have to keep in the mind about these like ethical, legal and social issues also. So when they are doing this project and when they are planning for this project, they have to keep in their mind that are we going to violate any moral principle? Okay, is this project is challenging any moral principle? They have to consider issues like privacy, bias, fairness, transparency and potential for harm, because we know that every good thing, every coin has two faces. So if we are like getting to know everything about the human genome and all those things, is like there is any harm to uh, any potential harm with that, is there like any issue regarding the privacy, biasness, fairness and transparency, that they have to keep in mind. Then talking about legal, does the project comply with all applicable laws and regulation? Example, data protection, intellectual property, accessibility standard. So they have to also keep in mind that this project, comply with all applicable laws and regulations. Means they have to keep in mind that this project is following all the laws and regulations which are, followed when people are doing some scientific project and all. Example are what? Like data protection, intellectual property, accessibility standards and all. What legal liabilities could arise? So like they have to also keep in the mind if in future they are going to any legal liabilities that could arise. So these all things they have to also keep in the mind. Then after talking about ethics and legal, some social issue which they have to also consider, like how will project going to affect the different groups of people, how people are going to react if they got to know about this project. Okay, will it exacerbate existing inequalities or create new ones? Means is it going to create any inequalities in the society? They have to keep everything in mind. What are the potential unintended consequences for the society? Means definitely they are not going like they don't have any intent of harming anyone or anything doing anything wrong to anyone, but is there any potential unintended consequences for society? Means like they are doing it for good cause, but is it any way that people will take it as like there are some potential harm, or is there any potential consequence which is not they wanted to have on people, but it may arise? So all those things they have to keep in mind. They have to address this ethical, legal and social issues also. So along with that scientific thing that they have to identify all the genes, the base pair of all this, base or pair order of this 3 to 10 power 9 base pairs in human DNA, everything. But they have to keep this ethical, legal and social issues also in their mind. They have to consider it, and they had considered it. If we talk about Human Genome Project highlight, what was the goal? Understand the human genome and its variation to improve what? Diagnosis, treatment, and prevention of disease. So, what was the main goal of this project? That if they will understand the human genome very properly, okay, then you know they and variation, then what will happen? They can improve the diagnosis, they can improve the treatment, and they can even prevent the disease. If we talk about timeline, it was a 13-year project completed in 2003. So, what was the timeline? It was a 13-year project which is completed in 2003. Started in 1990, completed in 2003. The 13-year project. It's important to remember the timeline. Who are the key players in the collaboration? It was US Department of Energy, National Institute of Health, NIH, Wellcome Trust, UK, Japan, France, Germany, China, and others. So from this slide, what you guys need to understand it and keep in your mind is that what is the main goal of this project? Its main goal is to understand the human genome and its variation. Why? To improve the diagnosis, treatment, and prevention of the disease. What was the timeline of this disease? That if we talk about the timeline, it was 13-year project started in 1990 and then completed in 2003. And these are some key players in the collaboration. Now, if we talk about the model organism, so many non-human model have been sequenced. This contributes to the broader biological understanding. It includes bacteria, yeast, C. elegans, a free-living non-pathogenic nematode, Drosophila, plants that is rice and Arabidopsis, and etc. So, what was the model organism? So, many non-human non-human model organism has also been sequenced, and this contributes to what? To understanding this biology at a broader space. It includes bacteria, yeast, C. elegans. What is a C. elegans? It is a free-living, non-pathogenic nematode. Then Drosophila, we know it's the fruit fly, and then plants like rice and Arabidopsis. Now, what was the impact of this human genome project? So, if we will talk about it, so first is the revolutionary advancement in healthcare. So, what kind of revolution will come in the healthcare field? First, personalized medicine, tailoring treatment based on individual genetic makeup. So, after this human genome project, we can know what the DNA sequence, the genetic makeup of the human. So, it became very easy, not very easy, but it became more convenient to treat a person on the basis of their individual genetic makeup, and that's what we call personalized medicine, because that medicine, that treatment will be based on that people, that individual genetic makeup. Because we know that many people react, or uh, react to these diseases, no, to the medicines differently. But if we know that what is the genetic makeup of that person, we might give them the medicine which they can have properly, and there is no side effect and all. So this human genome project can make a very advancement in personalized medicine. Then improve diagnostic. With this, we can faster and more accurately detect the disease. Then gene therapy even we have studied in biotechnology chapter. So gene therapy, we know, but it no, like by knowing this human genome project, it can also help in gene therapy. It has potential to correct the genetic defects. Drug development, targeted therapy with fewer side effects. As we know, as I told, because what now we know the individual genetic makeup. So, if we able to know the genetic makeup of the individual, so it will become very easy to make a target therapy for them, because we know that okay, its genetic makeup is like this, so it is going to react to this medicine like this, or it is going to be more effective for this patient. So, what it will do? It will make targeted therapy with fewer side effects. So, this is the drug development. Disease understanding, uncovering genetic basis of disease like cancer. So, like in disease like cancer, we know about there are many oncogenes, there are many tumor suppressor genes. So if we will understand this whole, if we got to know about the all genes and all sequences, so what we can do, we can uncover this genetic basis of disease like cancer. We will understand that disease very better. For example, like even in cancer, as I told, there are many oncogenes and tumor suppressor genes. It might be possible, and it's like it happens that in a single patient there are many genes that has been mutated, so that we can understand better, that we can understand disease better, that if that, for example, if there is a cancer patient, and it is like divided into grade 1, grade 2, grade 3, grade 4, and if it is like that, that grade 1 person has fewer genes which is mutated, and grade 4 has many genes that has been mutated, that's like how we can understand the disease. Okay, so for, if this uh, genes will get mutated, so it will do progression of this disease. So by this we can understand the disease. So this is the human genome project impacted on in healthcare. Then other is the improved understanding of human biology, revealed genetic basis of disease, enabled identification of genes which link to hereditary disorders and increased disease risk. So what can happen, like how it can help? So it can reveal the genetic basis of disease, as in, so there are many, it will enable us to identify genes which is linked to inherited disorder. So there are many genes, okay. For example, okay. So for example, there are two parent, okay, male and female, and both has some gene, and that gene is like heterogene, means one is dominant, one allele is dominant and other is recessive. And for mother also, one gene is dominant and other is recessive. But to the, when they will give birth to the child, that child has a particular disease, and but we never know like why that disease happened. It is an inherited disorder. So what we can do, if we will understand its whole genome sequence, then we will get to know, okay, this gene has recessive alleles, and that recessive alleles has come from parents, because that parents were hetero, has, you know one dominant allele and one recessive allele. That's why that phenotype hasn't been shown in parents, but when it comes to the child, he or she is showing that disease. So from this we can enable the identification of genes which is linked to inherited disorder or even increased disease risk. For example, if that one that parent only has one child who has this disease, another has, again just like parents, one dominant allele and another recessive allele. But we know that if that person is also going to marry to a other person who has that kind of gene only, then we can know now that their progeny now going to have this disease. So it will make us understand, okay, so these are the genes which is linked to inherited disorder, and this can increase the disease risk. So this is a very good thing, and this is the very, you know good application of this human genome project. Then advanced personalized medicine, as I told you previously also, because now once know the one will know the this genetic makeup of the person, it will become easy for giving the treatment, pave the way for tailoring treatment based on individual genetic profile. Then enhance drug development, facilitate the discovery of new drugs target and improve drug design. This will also help for the discovery of new drug target and improve drug design. Means that drug is going to target what, and therefore we can improve even drug design. Then expanded knowledge of human evolution, provided insight into human origin, migration pattern and genetic diversity. So by this project also, what we can do, we can expand our knowledge of human evolution, like what is the human origin, what was like migration pattern and genetic diversity. Then improved diagnostic tool led to the development of more accurate and efficient genetic tests for disease diagnosis and screening. So what other things in which it has contributed, that it has led to the development of what, more accurate and efficient genetic test for disease diagnosis and screening. Okay. So then come the application in agriculture, energy, and environmental remediation. So it has no direct contact with this human genome project. Now here we're talking about the application in agriculture, energy, and environmental remediation. If we talk about agriculture, crop improvement, identifying the gene for disease resistance, yield, and nutrition value. So if we will do that same sequencing for plants, so what we can do, we can identify the genes for disease resistance, the yield, and nutritive value. Then pest control, we can develop targeted environmental friendly pest control strategies. Now if you talk about energy, so energy, what it can do in energy, it can help in biofuel production. Means what we can do, we can engineer microbes for efficient biofuels production. Biomass utilization, we can optimize enzyme for breaking down biomass for energy. Then if we talk about the environmental remediation, so it can help in bioremediation, means identifying and engineering microscope to degrade pollutants. Then environmental monitoring, they can develop DNA based tools for monitoring environmental health. So these are some applications in agriculture, energy, and environmental remediation. So now comes the main part, that what is the methodology for human genome project. So first, the method involved two major approaches. First, identification of all genes, and second, home genome sequencing and annotation. So the method involves two major approach. First, they have to identify all the genes, and then they have to do the sequencing and annotation of the whole genome. So if we talk about the first, identification of all genes, so this focused on identifying all genes that are expressed as RNA. The RNA referred to as express sequence tag, those RNA. So first method is what is for identifying all the genes. This like this mainly focus on identifying all the genes that are expressed as RNA. Then those RNA referred to as express sequence tag, EST. These are the important terms that you guys need to remember. That express sequence tag. What is the express sequence tag? These are they refer to those RNA, okay, that they have that they want to identify, that the gene that they want to identify, and that gene is doing what is expressed as RNA. So those RNA is called express sequence tag. Then second method was whole genome sequencing and annotation. This included sequencing the entire genome, both coding and non-coding regions. Means it doesn't depend affect like whether that part is coding or non-coding. In this, like they have to sequence the entire genome. After that, function was assigned to each part of the genome called sequence annotation. So after they will sequence all the entire genome, then they will do, function was assigned to each part of the genome, okay, and that is what they call sequence annotation. Means giving assigning function to the part of genome, that's what they call sequence annotation. So these are the two approaches. Now if you talk about the what is the DNA sequencing process overview, so first is the DNA isolation and fragmentation. Total cellular DNA is isolated and broken down into smaller random fragments. This is necessary due to the limitation in sequencing very long DNA strands. So definitely if they want to sequence the DNA, first they have to do what? They have to isolate the total cellular DNA, and then they have to break them in smaller and random fragments. Why it is necessary? It is necessary because it's not possible to, there are many limitations to sequence a very long DNA strand. That's why they have to break them into smaller and random fragments. Now, let's talk about then cloning and amplification. DNA fragments are cloned into specialized vector, that is BACs or YACs. Bax we know bacterial artificial chromosome, or Yax is yeast artificial chromosome within host organism, that like will be bacteria or yeast. So this cloning amplification thing we have studied in detail in biotechnology chapter. So now what they did, they they have cloned that DNA fragment into specialized vectors. We have studied about these vectors, you remember? And which vector? The vector they have used is Pax or Yax. Within the host, here what are the hosts? Here the hosts are bacteria or yeast. Cloning lead to the amplification of each DNA fragment, making sequencing easier. So what did this? Why this cloning step was important? Because this cloning lead to the amplification of this DNA fragment, and because of this amplification of DNA fragment, the sequencing become easier. So this is a representative diagram of human genome project. So what they are showing that there is a this girl. Then they have taken their cell, they have taken out their DNA, that whole chromosome, then DNA. Now they sequence the DNA. Now they have database of the sequencing. Now it comes sequencing. First, what they did first, they have isolated the DNA and broken it into fragment. Then they have cloned that DNA fragment into vectors within the host organisms. Vectors are Bach or Yak, and host organisms are bacteria or yeast. Now it's time for sequencing, the main process, sequencing. Fragments are sequenced using automated DNA sequencer. Scientists here, the sequencer uses the Sanger sequencing method, which is developed by Frederick Sanger. It relies on the random incorporation of dideoxynucleotide triphosphate, that is DDNTP, during DNA replication. This incorporation halts the process and allow for identification of each nucleotide in the sequence. So now after the DNA isolation and cloning, what will they will do? They will sequence. How those fragments are sequenced? They are sequenced using automated DNA sequencer. Here sequencer are termed, they have termed sequencer whom? Scientists. Scientists use what? Scientists use Sanger sequencing method. And this Sanger sequencing method, who has developed the Sanger sequencing method? Frederick Sanger has developed this method. So now I'll tell you the summary which you guys need to know. You guys don't need to go into very much detail like what is totally properly Sanger sequencing method, but I will give you a gist. So what is this Sanger sequencing? So this Sanger sequencing is a method for doing what? For determining the precise order of nucleotide in a DNA molecule. This is what sequencing do. It is a method for determining the precise order of what? Nucleotide in DNA molecule. It relies on use of what? It relies on the use of dideoxynucleotide, that is dideoxynucleotide triphosphate, that is what? DDNTP. Now, what is special about this DDNTP? So, this lacks a 3' hydroxyl group, which is necessary for DNA polymerase to add further nucleotide. So, this DDNTP has what? It lacks the 3' hydroxyl group necessary for DNA polymerase to add what? Further further nucleotides. So you guys can see from here, if you guys remember of the DNA structure, so what was that? There was a deoxy, like why we say deoxynucleotide? Because there was an absence of OH group at 2'. Remember? But in this, there is even a lack of OH at even 3', and that's why it is called what? It is called dideoxynucleotide. Why it was important? It was important for DNA polymerase to add further nucleotide. We have studied in replication process that how this DNA dependent DNA polymerase is important for adding further nucleotides. Now, because they lack this, so now what will happen? This causes chain termination during DNA synthesis, producing DNA fragment of various length that can be analyzed to reveal the sequence. So this was the concept behind this. You guys understand what happened? So in this, they have used this dideoxynucleotide triphosphate. Why? Because it don't have OH on even 3 dash. They don't have OH at the 3 dash end, which is required for what? For DNA polymerase to add further nucleotide. Now, because they don't have this OH group on the 3 dash, this will cause chain termination during DNA synthesis. And what will happen? So now it will produce DNA fragment of various length that can be analyzed to reveal the length. So this is the basic concept of Sanger sequencing. So let's talk about the, like, I'll again talk about what? The principle. If we talk about the principle, DNA sequencing involves determining the order of what? Nucleotides in DNA molecule. The Sanger method uses DNA polymerases which add nucleotide to a growing DNA strand one at a time by forming a phosphodiester bond between 3 hydroxyl group of the last nucleotide and 5 phosphate group of the incoming nucleotide. This dideoxynucleotide, okay, what are they? They are the modified nucleotide that lack the 3 hydroxyl group. When a DDNTP is incorporated into the growing DNA strand, it terminates the further elongation because another nucleotide cannot be added because DNA polymerases needed that 3 OH group, okay, to function. So it terminates the further elongation because another nucleotide cannot be added. So if we talk about the process, so in a Sanger sequencing reaction, a template DNA molecule is mixed with DNA polymerase. Regular deoxyribonucleotide means regular DNTPs, okay, and a small amount of specific DDNTP. Okay, for example, DDATP, then DDGTP, DDCTP, like this. Okay, the reaction is carried out in four separate tubes, each containing different DDNTP, one for each base, ATCG. As DNA polymerase synthesize new DNA strand, it will occasionally incorporate a DDNTP instead of a normal or regular DNTP, causing the strand to do what? Causing the strand to terminate. This result in a set of DNA fragment of various length, each terminated by a specific DDNTP. The fragment are then separated by size using gel electrophoresis, which you guys can see in this figure, because I told you there will be like four tubes, each will have their specific DDNTPs, okay, for A, T, C, G. And what will happen? As the DNA polymerase will synthesize new DNA strand, occasionally what will happen? Instead of a DNTP, a DDNTP incorporate, causing the strand to terminate, and that's why it result in what? DNA fragment of various length that we can see, each terminated by the specific DDNTP. It can be ATCG. The fragments are then separated by using what? By using gel electrophoresis, with the smaller fragments migrating faster and the larger smaller. Now, by analyzing the length of the fragment and the DDNTPs that terminated them, the DNA sequence can be determined. So this is the basic of the, this is the basic principle and process of the Sanger sequence. So I don't think like in NCRT they have the whole process or principle. I have just told you this for your knowledge. So the main thing you have to make remember is that Sanger sequencing method is used. It was developed by Frederick Sanger. Okay, it relies on incorporation of what? Dideoxynucleotide triphosphate. Why? Because it has not lack 3 dash OH group. And why this 3 dash OH group is important? Because it enables the DNA polymerase to add nucleotide during replication. Okay, but if it is not there, it will terminate it. So this is the whole Sanger sequencing thing. Then sequence assembly. Now, how the sequence assembly will happen? So, okay, one thing I have to add, which I haven't added in previous slide, that this now like in this modern Sanger sequencing, people often use this DDNTPs but they are fluorescently labeled, means they have fluorescent label. Okay, fluorescence label. That's why you can have guys have even seen the figure that it was like showing green, pink, and all those color because it was fluorescent label, where each base is labeled with a different color, and this allow all four reaction to be carried out in a single tube. So we don't have to make four test tube. It can happen in the single tube because now, because it is fluorescent label, we can differentiate what it is. Okay, and the fragment are then separated by capillary electrophoresis, and the detector read the color of the fluorescent tag as each fragment passes. Okay, so this is about this. Now what will happen after that? Then there will be the sequence assembly. So what happened in this, overlapping region within the sequence fragment are identified. So there will be definitely some overlapping regions within the sequence fragments, and that are identified. Then these specialized computer programs are used to align and assemble the overlapping sequence into complete genome sequence. So what will happen after, when they have identified this overlapping regions within the sequence fragments, these specialized computer programs are used to align and assemble the overlapping sequence into a complete genome sequence. So this is what we call genome assembly, and this figure I have explained to you. Then there will be annotation and chromosome alignment. So the assembled sequence is annotated, means identifying genes, its regulatory elements, its function, and all those things. Thus, assembled sequence is annotated. The sequence are assigned to their respective chromosome. For example, the sequence of chromosome 1 was completed only in May 2006. So they are saying what, after that process, assembly process, what they will do the annotation and chromosome assignment, means this the assembled sequence is annotated, means they have identified the genes, regular elements, and all, and then the sequence are assigned to their respective chromosome. For example, if we talk, the chromosome was completed only in May 2006. Mapping the genome, it is a very challenging task. Genome mapping, if we talk about, it is a process of identifying and recording the location of gene and other important landmarks on a chromosome. These maps were created using information on polymorphism of restriction endonuclease recognition site, repeated DNA sequences known as microsatellites. So what they are saying, so in this they are saying, so mapping the genome is like whole genome is very difficult. Okay, and what is this genome mapping? It is a process of identifying and recording the location of genes and the other landmarks on the chromosome. Okay, it is very difficult. So, how these maps were created? These maps were created using information of polymorphism of restriction endonucleases recognition site and repeated DNA sequence known as microsatellite. So they have what? They have like these uh, the chromosome have DNA repeated DNA sequence, what they call microsatellites. So this became important in doing what? In making this and creating this maps, this genome mapping. And the second is, so these maps were created using the repetitive sequence which we call microsatellite. And second, this information on polymorphism of restriction endonuclease recognition site. And this refers to what? This refers to variation in the DNA sequence that are recognized and cleaved by restriction endonucleases. Okay, and these are the other things that these variation are known as what, this RFLP that we will talk I think later. But yeah, so these are the what? These are the variations in the DNA sequence that are recognized and cleaved by the restriction endonucleases. And I think you guys remember what is this restriction endonucleases, which can cut the DNA at the specific point. Okay, so these two are used for creating map. Then comes the salient feature of human genome. So this is very important. Like, what are the key finding of human genome project? So if we talk about the key finding of human genome project, so what are they? So they are the human genome. First is the genome size. They got to know that the human genome comprise of 3164.7 million base pairs. Gene size, if we talk about, so the average gene length is 3000 bases with significant variation. The largest known gene, the dystrophin, spans 2.4 million bases. So see, this is not something that you guys need to, understand, or like you guys need to understand as in these are the some things that you guys need to remember, that what is the, human genome comprise of this much billion base pairs, what is the average gene length. So these are important things that you guys need to remember. Gene number, the total number of gene is estimated at 30,000. This is significant lower than early prediction of 80,000 to 1,40,000 gene. So total number of gene which came out to be like is 30,000, but they have predicted that it would be like from 80,000 to 1,40,000. Then sequence similarity. So humans share 99.9% nucleotide based sequence identity. So see. Then the unknown gene function. The function of over 50% of discovered genes remain unknown. Coding region, if you talk about, less than 2% of the genome codes for protein. Repeated sequence and its function. This from here you guys can answer NCERT. Question number 8a. So the stretches of the DNA sequence that are repeated many times are called what? Repeated sequence. A substantial portion of a genome consists of what? Repeated sequence. Means a quite amount of genome is consisted of this repeated sequence, a sequence where this DNA sequence are repeated many times. Sequence can be repeated from 100 to 1000 of times. These sequences are directly involved in coding, but are not directly involved in coding, but provide insight into chromosome structure, dynamics, and evolution. So means these repeated sequence, they are not directly involved in coding as in they do not code for polypeptide for protein, but they provide a insight into chromosome structure, its dynamics, and evolution. Then chromosome gene count. Chromosome 1 has the highest number of genes, that is 2968. The Y chromosome has the fewest, that is 231. Then single nucleotide polymorphism. It is a variation at a single position in the DNA sequence among individuals. Approximately 1.4 million locations with single base DNA difference have been identified in humans. So what is this single nucleotide polymorphism? So it is a variation at the single position in the DNA sequence among the individual, and approximately how many? Approximately 1.4 million locations with single base DNA differences has been identified in human beings. This information helps in locating disease associated sequences and tracing human history. So these are the important key features, key salient features of the Human Genome Project that you guys need to remember. Then application and future challenges. Application of DNA sequence. Future research focus. Deriving meaningful knowledge from this DNA sequence will be the primary focus of research for decades to come. Means even after DNA sequence, what they have to do, they have to they have to analyze the meaningful knowledge from this DNA sequencing. This is one of the most important thing. This will drive understanding of the whole biological system. Collaborative effort. This is a large scale task. It requires expertise, creativity. 10 of 1,000 of scientists across diverse disciplines in both private and public sectors globally are needed. Transformative impact on biological research. Availability of the complete human genome sequence may revolutionize biological research methodology. So these are the what? These are the application of the DNA sequencing. Like how it is going to affect the future research focus. Going to need the collaborative effort, because this is not that only 1 or 2% can do. Okay, this is going to impact has a huge impact on biological research. Then shift from gene specific to genome wide studies. Modern research use large scale systematic approach instead of focusing on single genes. Comprehensive analysis HGP enables studies of all gene within a genome, all transcript within a specific tissue, organ or tumor. The interconnected network of 10 of 1,000 of genes and proteins at their coordinated roles in life chemistry. So what is they are telling? In short, they are telling that what enables, like what HGP has able to do, it has able to make us to know all the genes which is present within a genome. Okay, all the transcript, all the mRNA that is present within a specific tissue, organ, or tumor. Okay, the interconnected network of 10 of 1000 of genes and proteins, and how they are coordinated in, like, normal life chemistry, normal life biology. Now, what was the summary of today's class? The summary of today's class like this: the Human Genome Project was an international research effort to sequence and map all human genes, that is the genome. Key features include 3 billion DNA base pairs, approximately 20,000 to 25,000 genes, and 99% of the identical genome between humans. Application include disease diagnosis, personalized medicine, genetic counseling, and evolutionary studies. Further challenges involve understanding gene functions, ethical concerns about genetic privacy, addressing genetic discrimination, and managing vast amount of genomic data efficiently. The project revolutionized our understanding of human genetics and continue impacting medicine. So these are some, like, summary points. And in today's class, in short, you guys have to remember, like, few things. Everything you can't learn, but the things you can't learn, like, number of the DNA base pairs, identical genome, SNPs, all those important things which I have, like, focused, that you guys need to remember. So let's start with the NCERT based question. The Human Genome Project was launched in which year? So we know it is in 1990. The Human Genome Project was officially launched in 1990 and was completed in 2003. Then, what percentage of nucleotide base sequence is identical in all humans? So the percentage of nucleotide base which is identical in all human is 99.9%, is exactly same in all people, only 0.1% create individual differences. Then the largest known human gene is, so the largest known human gene is dystrophin. Dystrophin is the largest known human gene. It contains 2.4 billion bases. Then, what percentage of the human genome code for protein? So it is less than 2%. So less than 2% of the gene is going to code for proteins. Rest include regulatory sequence and non-coding DNA. Then, which chromosome has the most genes? So we know that it is chromosome number 1 which have the most gene. It has 2968 genes. Most gene rich chromosome in human is chromosome 1. So next question: the Human Genome Project identify approximately how many SNP? So we know that the Human Genome Project identify approximately 1.4 million of SNP are been identified. What are SNP? They are the single nucleotide polymorphism, means the single, variation in the DNA sequence, single base pair. Which vectors were used in the Human Genome Project for cloning DNA fragment? Only BAC, only YAC, both BAC and YAC, or neither BAC or nor YAC? So we have studied both BAC and YAC, that is the bacterial artificial chromosome and yeast artificial chromosome, that is are the two which is used as vector. Any YAC, that is yeast artificial, both bacterial artificial chromosome and YAC, that is yeast artificial chromosome, these are used as vector. Then the average gene size in the human genome is what? It is 3000 bases. Average human genome consists of 3000 bases. The total number of base pair in human genome is how much? So it is 3164.7 million base pairs. The next question is, for what percentage of discovered genes are known unknown? So it is over 50% of the discovered genes whose functions are unknown. Significant research still needed. So if we talk about the interesting facts, so this HGP, that is Human Genome Project, is completed in 2003. It cost approx one base it took 13 years. It was an international collaboration. It mapped 3 billion DNA base pairs. Salient feature: 20,000 to 25,000 gene identified. 99.9% identical between humans, and only 2% codes for protein. Now, if we talk about application, what are the application of Human Genome Project? Disease diagnosis, personalized medicine, genetic testing, drug development. Challenges, if we talk about, so it was a very huge data for the data interpretation. Ethical consent, privacy issue, cost of implementation, these are the challenges. Here are some interesting questions that you guys need to answer. That The Human Genome Project found that 99% of the DNA is identical among all humans, yet we all look different. Explain this apparent paradox. Why was the discovery of only 30,000 gene instead of expected 80,000 to 1,40,000 considering surprising? And what does this tell us about genetic complexity? Then, less than 2% of the human genome codes for protein. What might be the purpose of remaining 98% that was once called junk DNA? Then next question, how did the use of BAC and YAC help solve the massive puzzle of sequencing the human genome? Explain their role in the process. Then question number 5, the Human Genome Project dedicated a portion of its budget to studying ethical, legal and social issues. Why was this consideration so important for a scientific project? Thank you. So yeah, today's DPP. Question number first question says, what was the approximate total cost estimated for Human Genome Project at the beginning? So it was what? It was 9 billion US dollars. Because for one base it was $3, and then they multiplied it to 3 to 10 to the power 9 base pairs. So it was that. Then which chromosome in human has the fewest gene according to the Human Genome Project? So we have studied it was chromosome number Y. Then, what percentage of the human genome codes for protein? So, we know that less than 2% of the human genome codes for protein. Then, which of the following was not a goal of Human Genome Project? So, address ethical, legal, and social issues. So, we know that it is one of the goal of Human Genome Project that they have to address the ethical, legal, and social issues. Then, to determine the sequence of 3 million base pairs. Okay, this was also the goal of the Human Genome Project. Then develop methods for human cloning. So this was not the goal of Human Genome Project that they have to develop method for human cloning. No. An improved tool for data analysis. Yeah, this was the goal of the Human Genome Project. Then what are SNP in the context of human genome? What were the SNP? So it was single nucleotide polymorphism indicating base difference at one position. Yeah. Okay, so we have solved all the question. Let's submit it. So we can see it's all correct. So yeah. So now it's all for today class. If you guys have doubt, you guys can ask. Otherwise, we will meet in tomorrow class. Okay.